Welcome to the Pest and Parasites Master Level Training Module for the Texas Master Beekeeper Program. My name is Pierre Lau. By this level, you should hopefully have a general overview of some of the different types of pests and parasites that honeybees have to deal with. In this module, we'll be diving into a little bit more detail with these pests and parasites. Here's an outline of what we'll be covering today. First, we'll be defining and going over the differences of what defines a pest and a parasite. We'll then briefly summarize some of the pests and parasites that were covered in the previous exams, including tracheomites, wax moths, small hive beetles, and other arthropods, including ants, mantids, yellow jackets, and wasps, vertebrate pests, Nosema, and Varroa. However, we'll be going into a little bit more detail with Nosema and Varroa because of their economic importance in the honeybee industry. For each pest and parasite, we'll go through their biology and their life cycle. And that's because understanding their biology is one of the first steps for developing any kind of management technique. We will then quickly go over some of the common management techniques involved with each pest or parasite. Note that the management techniques that we go over is only a few of the commonly used methods, and then it's not a comprehensive list, and there is probably more that we are not going to cover today. So a parasite is known as an organism that lives on or in an organism of another species. They obtain nutrients from the host. On the other hand, a pest is an organism detrimental mainly to human concerns. Some examples for honeybees include wax moths, small hive beetles, and mammals and other insects. Within pests, there are primary and secondary pests. Primary pests directly reduces the overall well-being of a colony substantially, while secondary pests only cause excessive damage after the colony is stressed or weakened by other factors. These pests can be considered a nuisance to beekeeping. The definitions can be confusing because you can be a pest and a parasite, but if you are a pest, it does not necessarily mean that you are a parasite. The first organism that we're going to go over are tracheomites. Tracheomites are microscopic in size. They're about 1.5 times the diameter of a human hair, and they spend their entire life cycle within the trachea of the honeybees. The trachea are the breathing tubes of the honeybees. They can shorten the lifespan of adults by affecting their flight activity, resulting in a large number of bees unable to fly. The wings of the mite-infested bees are often disjointed, with the hind wings projected about 90 degrees from the axis of the body. However, absence of these symptoms does not necessarily mean that the bees are free from mites. Colonies that are severely infected might still appear normal until death during the winter. Infestation is typically the highest in the spring when the population is mostly older bees. So with the life cycle, I'm going to start off on the left-hand side. But within 24 hours of after the worker bees emerge from their cells, female mites actually collect within their trachea. And once the female mites get in the trachea, they can lay single eggs in the bee respiratory tract where their developmental cycle will occur. Each of the female mites can lay 5 or 7 eggs 3 or 4 days after entering the trachea, and the eggs require 3 to 4 days to hatch. After the eggs hatch, the adult mites, which are about 5 to 9 days old, emerge from the host trachea in search of a new host. Typically, the highest number of mites are found around 12-day-old bees, and mite levels decline after over 21 days old. After a female mite is mated, she leaves the trachea and climbs on to the tip of the body hair. The mite then attaches himself to the hair of a passing bee and then enters the trachea through the spiracles. And during this process, a mite has to complete this whole process of transferring bees within 24 hours, otherwise they will not survive. After infestation, adult mites will penetrate the tracheal wall with their piercing mouthparts and feed on the bee blood. Normal thoracic trachea are clear, colorless, and usually pale. However, when bees are severely infested, their trachea are darkened and it may actually appear black. So the main way to manage tracheomites involve the use of menthol. Menthol crystals were registered as a pesticide for tracheomite control in the United States in 1989. And how effective this product really depends on many factors, including temperature, the formulation of the menthol, so they can either be in crystal or pellet form, the dose that they're applied in, the colony, the whole size of a colony, and how long the colony is exposed to menthol crystals. These crystals provide a fumigant action that kills tracheomites without harming bees at certain temperatures. And these crystals actually evaporate at about 70 degrees Fahrenheit and will melt to a liquid state around 2 to 105 degrees Fahrenheit. The vapors that these crystals emit are heavier than air, and the best evaporation rate occurs at around 80 to 85 degrees. It should also note that menthol treatment should only be used during a non-nectar flow period and after the surplus of honey has been removed, and that's really mainly to prevent menthol-flavored honey. The next pests that we are going to go over are wax moths. Wax moths are holometabolous insects that tunnel through the beeswax comb and consumes comb-containing resources. Since they typically will not kill a colony by themselves, they are considered to be secondary pests. The adult female lay eggs in protected crevices near food sources, and the eggs often appear to be creamy and to be a creamy white color. How long it takes the eggs to hatch really depends on the temperature that they're experiencing. And that's because warmer temperatures will lead to faster development for all life stages, but the eggs will typically take 5 to 8 days to hatch. Once the eggs hatch, the larvae development can take between 1 to 5 months at an average of 6 to 7 weeks. The larvae undergo 7 molts, with most of the growth happening within the last 2 instars. As I mentioned earlier, the larvae tunnel through the beeswax comb, spinning tunnels of silk, which they often cover in feces. And this larval stage is actually the only stage where the wax moth eats. The larvae typically consume comb-containing bee brood with honeybee larvae and pupae, pollen, and honey. And the larvae actually prefer comb containing this brood or pollen over fresh comb. As they mature into the pupal stage, the mature larvae will spin silk cocoons around themselves prior to pupating. Cocoons are usually white and have webbing to hold them in place. The cocoons are covered in feces and other debris, and the pupae can take up to 2 months to mature, but the average time to adult emergence is about 37 days. Adults live approximately 1 week and are most active at night. Mating typically occurs within honeybee hives, and males attract females to mating sites with ultrasonic signals. Females also lay their eggs at night. During the day, adults remain hidden in trees and bushes near hives. Wax moths are also considered secondary pests of honeybee colonies because they only become a nuisance to colonies that are weakened already by another factor, such as a failing queen, poor nutrition, primary pests like varroa, pathogens, and small hive beetles. The best way to manage wax moths is really to maintain strong colonies. A strong colony will typically be able to defend themselves pretty well against wax moths. Wax moths are usually the most damaging when there is stored comb that is not protected by resident bees in a colony. If you do happen to find yourself with a wax moth infestation, such as a picture depicted on the bottom right, extreme heat or cold will kill all life stages of wax moths, including eggs that may be hidden from view. Temperatures of above 114 degrees Fahrenheit is sufficient for killing the wax moths, and that's perfect because the wax comb melts at around 119 degrees Fahrenheit. Small hive beetles are holometabolous insects that have distinct life stages. They're responsible for the fermentation of hive products, such as honey, associated with feeding larvae. This is likely due to the specific yeast associated with the small hive beetle. Honey damaged by small hive beetles is rendered foul and unfit for human consumption. Colonies heavily infested with small hive beetles may abscond, although the number of adults needed to actually get them to abscond is usually very high. However, small hive beetles are still considered to be secondary pests. I'm going to start talking about the life cycle on the bottom left-hand corner of the cycle on the right. But upon emerging from the ground where they pupate, the adult small hive beetles will search for honeybee colonies using mainly olfactory cues. They can possibly use odors from various hive products as well as the honeybee alarm pheromone. Once they locate and enter a host colony, adult small hive beetles seek out cracks and crevices where they hide from bee aggression. Bees keep the small hive beetles confined to the cracks and out of the brood comb, where there's an ample supply of honey, pollen, and brood on which small hive beetles reproduce. However, small hive beetles will not starve in these corners that they're kept in because they have the ability to solicit food from bees using their antennae to rub the bees' mandibles and inducing the bees to regurgitate their food. The mating behavior of small hive beetles is not too well understood. However, if they are allowed to reproduce, small hive beetles will oviposit directly onto the food sources such as the pollen or brood combs. They can deposit eggs in crevices or cavities away from the bees, and the female hive beetle may lay about 1,000 to 2,000 eggs in her lifetime. The majority of these eggs will actually hatch in about 3 days, but this really depends on the conditions. Humidity appears to play a pretty big factor for hatch rates because eggs are prone to desiccation if exposed to circulating air and if the relative humidity is below 50%. The newly hatched larvae immediately begin feeding on any type of food source that's available. Food sources can include honey, pollen, and breed brood. They do, however, have a preference for bee brood. Maturation time for larvae is generally variable, but is around 10 days. Once the larvae finish feeding, they leave the food source and migrate out of the colony to find suitable soil in which they pupate, and this is usually done at nighttime. Most larvae pupate within a meter of the hive, and the larvae burrow and construct a smooth-walled cell in which they pupate. The period of time spent in the ground pupating can vary depending on many factors such as the soil temperature, what the soil is composed of, and the moisture content. However, the majority of adults will emerge from the soil in about 3 to 4 weeks. And once they emerge, the life cycle will begin again, and the average cycle from egg to adult will usually occur within 4 to 6 weeks. To manage and minimize small hive beetles, it's very similar to wax moths where you maintain strong colonies and a clean apiary. Strong colonies can usually fend off small hive beetles on their own. One thing to note is that pollen supplements can induce a greater small hive beetle infestation because the extra food will facilitate a larger small hive beetle population. There are various methods of chemical control and mechanical traps that can be used, but I'm just going to talk about one example that's shown in the picture, and this is actually one of the more economically friendly ones, and it's the use of Swiffer wipes. By placing cut pieces of non-scented Swiffer wipes on top of frames, you can actually capture the small hive beetles using the fibers of the wipes, and this can occur within a matter of days. Here I'm just going to briefly talk about some of the other insect pests that honeybees may encounter. I'm first going to start with the obvious one of predation. Although this may ultimately not play a huge role in colony health, the added risk of encountering a predator is one of the reasons why the oldest honeybee workers go out to the environment. Research has shown that foragers could detect and avoid predators such as praying mantises based on the mantis's odor and visual appearance. Though a foraging worker ended up reducing their recruitment dancing by about 1.8-fold for a food source with the live adult mantis, even though they weren't attacked. Therefore, even though workers are not directly being attacked, they showed aversive behaviors for foraging, which can ultimately affect foraging for resources. Ants are also a pest, as they are known to raid hives for resources such as honey, nectar, and larvae. Although there is not much attention paid to this, it is actually a really common problem that many beekeepers report to have. Because of their size and their numbers, honeybees have little means of defense against ants, as they're usually too small to be stung. To reduce the opportunity of ants getting in your hive, different strategies have been employed by beekeepers, such as the one pictured on the bottom right-hand corner. The legs of a hive stand or a table can be surrounded by vegetable oil, water, or tanglefoot, which is a non-sticky residue derived from trees. However, this has to be constantly monitored because ants can find other paths of getting through. In addition, I want to bring up the possibility of a spillover effect of viruses from other insects to honeybees, such as ants. In New Zealand, the deformed wing virus complex, which is one of the viruses mainly transmitted by varroa mites, was actually detected in Argentine ants, which is a common invasive ant in the United States as well. There is much research in this field, but there can be many implications if ants can actually be a reservoir for honeybee diseases. In addition to insect pests, vertebrates can also be a pest for honeybees. Among the vertebrates, mice are the most common and troublesome for honeybee colonies. They mainly become a problem during the fall when the temperatures begin to drop because the beehives provide food in the form of pollen, honey, and bees, and protection from the cold. The mice can also destroy the frames and wax comb by chewing them to provide room to build their nest. Not only is this destruction of equipment a problem, but the odor created by the urine and the droppings can cause the bees to abandon a hive. These mammals can be kept out of colonies by reducing the size of an entrance as the weather begins to cool. Mice also nest in stored bee equipment, resulting in the same kind of damage. Therefore, care is also needed to keep these pests from the stored equipment. Another vertebrate pest of bees are bears. Bears enter a bee yard to feed on the honey and brood. The bears will usually affect 1 to 3 colonies a night, but will continue to return until all the colonies in the apiary have been destroyed. Electric fences are one solution against hungry bear, but depending on the bear's determination, the electric fence may not even deter him from getting to the hives and destroying them. Once a bear acquires a taste for honey, sometimes the best defense is just to move the colonies to another site. Finally, I want to talk about bee rustling. Bee rustling is on the rise as these attempt to steal colonies for monetary profit. It's unfortunate that this is a thing now, but measures should be taken to protect your colonies from thieves. Nosema is a microsporidian fungal parasite that causes nosemosis in honeybees. This condition is a primary pest that is about 5 micrometers in size. It is summarized as a disease that causes earlier foraging and pathological changes in their midgut epithelial cells, digestive and metabolic disorders, and malnutrition. Because of these reasons, it's considered to be a primary pest of honeybees. Nosema is an obligate parasite, meaning they cannot complete their life cycle without exploiting a suitable host. In this case, Nosema is completely dependent on the honeybee for energy. Here we are going to talk about why that is. As honeybees consume spores, the spores germinate and invade the midgut cells. When a spore germinates, it expels an internally coiled tube called the polar tube. The polar tube then infiltrates a host cell membrane, and the contents of the spores, also known as sporoplasm, are passed into the host cell. Once it's inside the cell, the sporoplasm begins to proliferate, and the resulting progeny, which are also known as vegetative states, procure nutrients from the host cells. As mentioned earlier, Nosema is dependent on honeybees for energy, and that is because they lack fully functional mitochondria, which produce large quantities of energetic molecules, also known as ATP. As a result, infections are frequently energetically costly for a host. In addition, Nosema vegetative states also likely undergo sexual reproduction, and the new spores formed can further propagate infections. Here, I have 2 species that are economically relevant to honeybees, Nosema apis and Nosema ceranae. Studies suggest that Nosema ceranae is potentially more virulent to Apis mellifera than Nosema apis, and that globally, Nosema ceranae was replacing Nosema apis. One likely reason is because Nosema ceranae originally infected Apis ceranae, the Asian honeybee, And then the host shift from Apis ceranae to Apis mellifera was a hypothesized driver between its alleged greater virulence. And that is because pathogens that successfully traverse host species barriers are sometimes more damaging to their new host than the host that they co-evolved with. Here we're going to talk about some modes of transmission. There are two main ways that Nosema is transferred. The first one is through trophallaxis, which is shown on the top left picture, and the second one is through the fecal matter on the bottom left. However, recent studies have detected viable Nosema spores in both the pollen pellets and royal jelly. As a result, it was recently suggested that Nosema can potentially infect honeybee larvae as well as adults. Now, to diagnose whether or not you have a Nosema infection, it's actually pretty difficult to do without the help of a lab. At the moment, only molecular techniques can determine both infection intensity and the species that's infecting your colonies. Furthermore, molecular techniques are more sensitive than light microscopy and can detect infection at very low levels as they amplify DNA from both the vegetative and spore parasite states. For Nosema management, fumagillin is an antimicrobial agent that is currently the only approved Nosema treatment for North America. When using this treatment method, be sure to follow the labeled directions and to give the full dose. Its effectiveness is dependent on the right dosage. The product may not work when too little is applied, and too much fumagillin can potentially be toxic to humans as a result of the residues left in the hive. However, many studies have found that an abundant and diverse pollen diet increases honeybee longevity to Nosema infection levels. This is likely to compensate the negative effects of Nosema hijacking their energy stores. However, the higher protein diet were also correlated to higher Nosema infection levels among workers. Despite the higher infection levels, there's still a net benefit of having a balanced diet. Since Nosema ceranae is more tropically adapted, germination is inhibited by low temperatures. However, cooler climates can mean that workers are limited in their ability to defecate outside the colony, which can lead to a greater infection level in the spring and the fall. Most of the information on Varroa Destructor is actually covered in Lance's IPM talk for Varroa talk in the advanced module. Therefore, I'll only briefly go into this topic, and you can refer back to Lance's presentation for more information. But Varroa Destructor was first reported in Southeast Asia, but since has spread globally. They are arguably the number one enemy of honeybees because they can carry diseases such as the acute Kashmir Israeli complex and the deformed wing virus. Because of their huge impact on the beekeeping industry and to colony health, they are considered to be a primary pest of honeybees. Like Nosema, Varroa are obligate parasites. Once again, that means that they require the honeybee in order to reproduce. Their life cycle involves two distinct stages, the phoretic phase and the reproductive phase. I will go over these two phases in more detail in the next two slides, but this image summarizes the life cycle of Varroa. I'm going to start off with the reproductive phase, where it is summarized into 5 sections: cell invasion, chemical mimicry, feeding, oviposition, and mating. The invasion of the brood cell occurs a couple of hours before a cell containing a bee larvae is sealed. This officially represents the beginning of the reproductive phase. Within the brood cell, the mite feeds on the bee and lays its eggs on the surface of the cell wall. Honeybees usually recognize their nest mates through the chemicals, and it has been shown that Varroa mites can actually mimic the chemicals a honeybee colony emits and modifies it according to the developmental stage of the host. Doing so reduces the chances of honeybees being detected and removed. Once a mite gets into the cell, they remain trapped at the bottom where the larval food is located at. They will then feed on a developing bee. As a result, newly emerged bees have less protein in their hemolymph and a drastic reduction in overall longevity. After 60 hours after the brood is sealed, the mites start laying eggs. The first egg is a male, and then subsequent eggs are females. Mating between the male, which emerges from the first egg laid, and the subsequent females takes place in the sealed brood cell. The mite will then feed on the bees until it is time to leave. Upon emergence, the mother mite and the offspring emerge and move on to search for a new host to begin the phoretic cycle, while the male mite will die in the cell. During the phoretic phase, mites hide between the abdominal sternites of a worker bee and consumes the worker's fat body and hemolymph. Mites prefer nurse bees to forager bees because of the chemical signature of forager bees. A forager bee's chemical signature has actually been found to have a repellent effect on the mite. By choosing a nurse bee, they can be led to a brood area where they can increase their reproductive success. However, if a forager is infested, it can actually facilitate the horizontal transfer of mites between colonies. In addition to drifting bees transferring varroa mites between colonies, a recent study showed that a varroa mite is capable of transferring from a flower to a foraging bee that visits the same flower. As a worker lands on the flower, the mite can actually detect them through the various cues and latch on to the bee. This behavior is considered to be risky for the mite, so typically only older mites do this because they are more desperate to do so. There are a lot of different methods for managing varroa, but first and foremost, it is important to monitor your varroa levels and ensure that they don't exceed the threshold. Two ways of doing this is through the use of a screened or sticky bottom board and by doing a powdered sugar shake. If the mite levels exceed the threshold levels, measures should be taken to control for varroa. These measures can include chemical control and/or integrated pest management or IPM methods. For more information about this, you can refer back to the Lance's advanced module. One topic that I do want to touch on is hygienic behavior. Hygienic behavior is considered to be a natural defense against varroa. Although it is not behavior specifically targeting varroa, it can lower varroa populations within a colony. Bees that are hygienic can detect many problems that affect brood, including varroa and cap cells, and remove the affected brood. Often, this behavior can lead to the death of the mite, thus lowering varroa populations. Hygienic behavior can also lead to increased grooming among individuals in the hive, and this behavior can ultimately remove mites that are phoretically attached to the worker. With that concludes the master module of the pests and parasites for the Texas Master Beekeeping Program.